This slideshow is going to look at the last learning objective in cells, looking at the significant organelles found in eukaryotic cells. So as you can see, in the first row, you're seeing stained images of the different components of the nucleus from eukaryotic cells. In the second row, the ER, the Golgi apparatus, and the lysosomes are part of the endomembrane system. Then you see important components listed below of the cell membrane and the cell structure with the cytoplasm, the microtubules, and the actin. We're going to look through each one of these in subsequent slides. So the nucleus is the cell's control center. It has all of the genetic material and also functions sort of like the brain and is the control center to tell each of the other components, the different organelles, what to do, how to do them, when to stop, and is also important in apoptosis, which is programmed death when it's time for the cell to be destroyed. So as we look on the upper image, you see a scanning electron microscope image of 3 different cells, and the nuclei of the cells have been stained blue. The nucleolus is the portion inside of the nucleus where ribosomes are produced. And so you see that the nucleus is the entire component. It would be the entire blue stained structure in the cells above. And the nucleolus is a significant component of the inside. And again, that's where ribosomes, the protein production units, are made. Chromatin is also found inside the nucleus, although it's not shown in this image. And the chromatin are thin fibers that house all of the DNA. And you can see in the blown up image, the stylized cartoon here, that the nuclear membrane has a series of nuclear pores, and these are proteins, protein channels, that are going to enable the RNA and the ribosomes to leave the nucleus to go out into the cytoplasm to perform their functions. So once you leave the nucleus, you're in the cytoplasm. That's a jelly-like substance that provides a little bit of support and protection for the organelles inside, and the cytoskeleton, which provides a far more significant component of the support. These are branches of proteins. Some of them are thick, others are thin, some are straight, some are branched and curved, and they act sort of like a roadway that enable the organelles and different cellular material to be transported throughout the cell. They provide structure and scaffolding for the cell and provide support, and in some cases can be used for movement of the cell, particularly in single-celled organisms like protists or in mobile and motile cells in a multicellular organism. So the cytoskeleton, as I said before, has 3 main purposes. It's going to provide the support and structure for the cell. It's going to act like a road system and control traffic and movement of components within the cell. And it's going to enable movement. You can see in the image on the left, protein microtubules have been stained so that you can see them. They're very densely structured around the nucleus and then kind of branch out from there, and so you can see where the plasma membrane would be, an outline that would kind of connect all the ends of these microtubules. And in the other image, you see actin, which is one type of protein filaments, in red, the microtubules in green, and then the nuclei are once again stained in blue. Of course, they are not a part of the cytoskeleton. So cilia and flagella, these are going to be essential for motion. In the images you see electron microscopy images of cilia on the surface of lungs and the flagellum on a sperm cell that's fused to an egg cell. So cilia, as we'd mentioned last week, can move fluid past the cell. This is going to be important to get the blood moving in the lungs so that the lungs are able to have the carbon dioxide and oxygen exchange. The cilia can increase the surface area and also get them moving so that you can get the carbon dioxide and oxygen going past, while the flagella are going to be responsible for moving the cell itself. The flagella is the individual tail on each sperm that's going to enable it to, swim, as we think of sperm as doing. The mitochondria are the cell's energy converters. What they're going to do is be responsible for taking the nutrients that we bring into our body, things like glucose and sugars, and transform those into ATP, which is the energy that cells are able to use. Cells cannot directly take in glucose, but they can use ATP, and these are two mitochondria within a lung cell. And if you look at a kind of stylized diagram of a mitochondria, you see that it too has this double membrane. It has one membrane on the outside, which is called the outer membrane, another one on the inside, which is the inner membrane. And if you notice, the inner membrane has all these cristae, all these crenulations. So the surface area of this inner membrane is much, much larger than the surface area of the outer membrane. And you're going to see in the chapter on cellular respiration that this is an integral component for the function of mitochondria. Within the inside of the inner membrane, we're going to have the matrix, and it's within the matrix that the ribosomes for the mitochondria are housed, as well as the mitochondrial DNA. And the space between the two membranes is just called the intermembrane space. And depending on the type of cell and how active it is, it can have up to thousands of mitochondria within it. And again, the function of the mitochondria is going to be converting sugars into cellular energy, ATP, so that the cells can function. So we talked last week about the endosymbiotic theory, and do you remember this, what we talked about? It is the hypothesis, beyond the hypothesis, the theory now, that certain prokaryotes with specific functions, such as the ability to convert sugars into large amounts of ATP or to provide photosynthetic capabilities, were engulfed by a larger prokaryotic cell and formed a type of symbiosis and eventually evolved into organelles within a eukaryotic cell. And some of the lines of evidence for this is that mitochondria, as well as chloroplasts, have their own DNA, separate from the DNA housed in the nuclei of each cell. The structure of the DNA, the mitochondrial DNA, is more like a prokaryotic cell's DNA than a typical eukaryotic cell. And there's a double membrane. Each of these organelles has two separate membranes. You saw on the previous slide, the inner membrane and the outer membrane. Having been engulfed could explain if the vesicle that engulfed the original prokaryotic cell remained, that could explain part of the double membrane. So if you think about it, it's interesting. We all have DNA from both parents, but we have more DNA from one parent than the other. And if you want to take a moment, pause the video, and think about it, who is going to be the bigger contributor, your mother or your father, and why? So pause the video while you think about it and start it when you're ready. Okay, so if you think about it, we know that you're going to need a large amount of energy for a sperm cell to swim. And so the mitochondria are absolutely jam-packed into the tail of a sperm, but an egg is going to be a lot less motile. It's going to have its mitochondria in the bulk of the cell, which is going to stay in place. When a sperm cell fuses with an egg cell, the tail actually disintegrates and only the head fuses, but the head has practically no DNA, mitochondrial DNA in it. And so the father's mitochondrial DNA is not given to the offspring, only his nuclear DNA. So your mother is actually a stronger DNA contributor than your father. Now moving on to a different organelle, lysosomes function like a garbage disposal. They're pretty much just these acid-filled garbage disposals. They have up to 50 different enzymes in them so that they are capable of degrading, breaking down pretty much anything that becomes a waste product in the cell. They take this cellular waste and digest it. Some of it can be returned out of the lysosome and into the cell for reuse, and some of it is just packaged up into a vesicle and removed from the cell. Just like any other function or portion of the cell, this lysosomes can malfunction, and a very serious genetic disorder is called Tay-Sachs. It is common, or more common, in Ashkenazi Jews than any other segment of the population. And with this, the lysosomes are lacking a large number of the enzymes that they are supposed to have, and it is almost always fatal by age 1, because without these enzymes, the lysosomes are not able to remove the cellular waste, and essentially the cells just fill up with metabolic wastes and the child dies. It's a terrible, tragic disease. So if you think about the organelles which we've gone over, which cellular component would be responsible for the classic shape of a red blood cell, this biconcave disc? And hopefully you said the cytoskeleton, and if you did, you'd be correct. Okay, moving on to more components, more organelles. The endomembrane system is comprised of the rough endoplasmic reticulum, which is section 3 here. It is lined with these little purple-blue ribosomes. That's what makes it rough. The smooth endoplasmic reticulum is the portion over to the side. It's a bunch of discrete sections that does not have ribosomes coating it. The Golgi apparatus is over here. These are separate little envelopes that are going to be important for packaging up materials to leave the cell. And then we have vesicles, like number 6 that goes off to number 7 here. Each of these are going to produce and modify molecules that can leave the cell to go to other parts of the organism. And the smooth endoplasmic reticulum, or the smooth ER, is also going to be responsible for breaking down toxins. If you have chemicals that your body needs to get rid of, whether they're purposefully or not ingested, but things like drugs and alcohol will also be broken down here, and then other cellular byproducts, other cellular waste can be processed there prior to going into the lysosomes. So if we look at the functions of the endoplasmic reticulum, you can see it is all this crenulated portion, and off to the bottom right is the nucleus. The endomembrane system is found immediately adjacent to the nucleus, and it's going to synthesize and export proteins. In the rough ER, so all those ribosomes are going to be essential for the protein manufacture, and lipids as well. The smooth endoplasmic reticulum is going to make most of the lipids, including the sterols and cholesterol, as well as some carbohydrates. So as we look at the rough ER, the endoplasmic reticulum, that's the one closest to the nucleus. You see the nucleus in purple, the rough endoplasmic reticulum, or rough ER, in sort of this red, and you see it's dotted, coated with all of these ribosomes. Again, the rough ER is going to modify proteins, produce and modify them so that they can then be packaged in a vesicle and sent to other parts of the body where it's needed. The smooth ER is going to synthesize lipids, fatty acids, phospholipids for production of other cells, as well as sterols, including cholesterol. And it's going to be part of the detoxification system for things like alcohol and drugs. So the liver, which is going to be responsible for a lot of the alcohol breakdown, is absolutely packed with smooth ER, and this isn't always a good thing. You will tend to have more smooth ER if one consumes a large amount of alcohol or has a drug problem, and it can desensitize you as it becomes better at metabolizing the drugs or alcohol because you have more of it. Your tolerance builds up, and so for someone who is an alcoholic, they'll need to consume more and more alcohol or consume more and more drugs to get the same, I'm air quoting, benefits from what they'd had before, to get that same high or fix. And so that can have very serious consequences for someone who's afflicted with one of these disorders. So pause for a moment as you think about these three questions, and when you're ready, come back and I'll answer them. Okay, so which would have the smoothest endoplasmic reticulum, or the most smooth endoplasmic reticulum? And that would be D, the liver cells of an alcoholic. For a bodybuilder, the portion that might become hyperdeveloped would be the rough ER, because they're going to be needing a large amount of proteins and muscle mass, and so the rough ER is going to get its workout, so to speak. And I'd mentioned before that long-term drug use or alcohol use, the smooth ER becomes better and better at detoxifying, at breaking down the drugs and alcohol that are coming into one's body. And so even if you're taking, say, a prescription medication now instead of a drug or an illegal drug or alcohol, your body is so able to process it that you might need a larger amount of that medication than somebody else, even though you'd never encountered that medication before. So as we move on to another component of the endomembrane system, we have the Golgi apparatus. This is not connected to the rest of the endomembrane system, to the smooth or rough endoplasmic reticulum, but it's a series of flattened membranes. Each one is called a Golgi body. So if you look at this sort of stylized diagram, we have, going from left to right, 1, 2, 3, 4, 5 separate Golgi bodies that are making up this Golgi apparatus, and each one of them will be responsible for processing molecules, the proteins that the rough ER or the lipids that the smooth ER had made, and package them up and provide them in little vesicles that you can see budding off here to be essentially shipped out of the cell to be used elsewhere. They will also produce carbohydrates. They can do carbohydrate synthesis by themselves, and other complex polysaccharides that are found within the plasma membrane. So those little whiskers that we saw before are largely produced and then processed for shipment elsewhere in and by the Golgi apparatus. So if we kind of synthesize all of this and look at the endomembrane system together, we're going to first have these transport vesicles that are going to bud off from the endoplasmic reticulum, either the rough or the smooth. Here we happen to be looking at the smooth ER on the top right here. The vesicle is going to take whatever it was that was just made, the lipid, and bring that, transport it to the Golgi apparatus, where you can see that the two cells, the two organelles, the vesicle and the Golgi apparatus wall, are fusing together, just like you saw with any other form of endocytosis. Those secretory proteins are going to allow that to connect, and the lipids that just moved over are going to be sent into the Golgi apparatus. It can kind of move through one of those and will be put off into a second vesicle. That the first vesicle was programmed to be able to attach to the Golgi apparatus. The second vesicle, once it has processed, is going to be able to attach to the plasma membrane, where then exocytosis can take place, and that lipid or that protein can then be released into the rest of the body for use somewhere else. So this is going to be an activity that we're going to work on in class. If you are in the face-to-face class, if you're in the hybrid class, I would like you to bring your analogy to class so that we can look over it together and group brainstorm to kind of look at the strengths and weaknesses of each of your models. Now, so far, everything that we had looked at are found in in animal cells, plant and animal cells. But now we're going to look at some organelles that are specific only to plants. One of them is the cell wall, and you can see that it's these cells and the plants are always drawn as sort of a box or a rectangular shape because the cell wall gives them a very fixed, rigid structure. And that's really their purpose. The cell wall provides structural strength, increased water resistance, right? If you're a plant and you can't go to a water source, you want to make sure that you house that water as well as you can. And a little bit of protection from things that might try to eat you. And the plasmodesmata are going to allow water and nutrient transport between the cells. We'd mentioned those last time, but the plasmodesmata are going to be these kind of large openings that are going to allow for very easy fluid transport between the different cells of a plant cell of a plant. The vacuoles are present in some animals, but not many, and they're never as large. But you can see in the bottom image that the pink pigment is being stored in this vacuole of a spiderwort, and that vacuole is absolutely immense. It's, in some cases, the largest organelle in the cell, even larger volumetrically than the nucleus. And vacuoles are going to have a series of different functions, from storing nutrients to holding waste materials, accumulating poisons, so your poison ivy is going to have to hold that poison somewhere, and it's going to tend to be in the vacuoles, and can hold pigments to do things like attract pollinators, say, look at me, I'm all pretty, right? And then the bees or whatnot come over and help with pollination. And when they're nice and filled up like this, they can provide extra support to keep the plants remaining upright. And so again, these central vacuoles can have a series of different functions. They can store nutrients, store waste, sort of like the lysosome in an animal cell. They can store poisons, either bad smells or actual toxins to avoid predation. They can attract pollinators by storing hormones or just pigments for color to attract things like moths or bees. And they can, through water turgor, provide actual physical support. And the chloroplasts are going to be the site of photosynthesis, the area where sunlight is converted to sugars. And there are a few structures that are important, and we're going to really focus on these when we look at photosynthesis in a few chapters, but I'm providing the vocabulary for you here so you can get used to it, and then we'll come back to it in a few chapters. And so once again, that endosymbiotic theory, chloroplasts resemble photosynthetic bacteria in terms of their size and in terms of the type of DNA that they have. And they too, like the mitochondria, have a dual outer membrane. So think about this for a moment. Which one would not be found in a eukaryotic cell? And of course, hopefully you said number 2, a DNA without a membrane is only found in prokaryotic cells.